record. Let's go back to the board. Where's the board? Where's the board? Yeah, here. We said that we want to make a scale. For neutral, who remember what's the concentration of H plus for neutral? Water. This is only in water, by the way.
7.
0.0000001.
10 to the power of -7.
6
10 to the
zeros,
power of
7
1.
times.
-7.
Or you can say the scientific number 1 times 10 power minus 7.
10 minus 7.
Excellent. What about if you choose a point below than neutral, acidic? Who can give me example of acidic concentration H plus?
What?
Yeah, give me example. One of you.
HCl. HCl in water.
No. Yeah, solution of HCl in water. But give me, what do you expect? Expect a concentration of H plus in this solution. Give me a number, expected number. 0 point. It's supposed to be greater than the water.
0.0.
10 to the power of -5.
5. Me.
5,
4.
6.
10 to the power -5.
4.
Hands.
4.
Give me hands.
4.
Or write it in chat. Write it in chat.
Sir, I couldn't
Write in
understand.
chat. Write in chat. Expect a concentration. Expect a concentration of H plus in any acidic solution. Acidic solution. What is the concentration of H plus? Expect. Is it greater than this number or less? And expect one, yani write one in the chat. Don't give me scientific number. Give me concentration number, like the blue one, 0.00 blah blah blah, like this. Hurry up, guys. Till now, I didn't receive any correct answer. Oh, yeah, I just receiving. Faizan, give me, how many zeros there? 1, 2, 3, 4, 5, 6, 7. No, Faizan, you gave me, okay, your answer, Faizan, not for acidic, it's for basic. Balaji, correct. Farhan, wrong, because Farhan gave me zeros only. What zeros? So nothing there. Abdullah Malik, correct. Tijan, correct. Muhammad Abu Ghandar, correct. Farhan, correct now. Vincent, wrong. Vincent, this number you gave me for basic, not acidic. Ali Abbas, give me, Ali Abbas, 1, 2, 3, 4. Ali Abbas, you give me neutral number, Habibi. It's for water. This is the neutral. I want one for acidic. Expect number for acidic. Mustafa, wrong. Mustafa, this is for basic. Ali Abbas, wrong. For basic. This one you gave me. I want one for acidic. Expect a concentration for acidic. Concentration of H plus. Guys, we said in acidic, the concentration of H plus is greater. Here it's greater. Remember, the concentration of H plus in the acidic is bigger, greater than in neutral. So the number should be bigger. 0, 0, 0, 1. Example. Okay. This number smaller bigger, sorry, greater than the H plus concentration in water. Okay. If I want to convert that number I just wrote for acidic, can you tell me what is the scientific number for this number? Quickly, scientific number. Convert it to scientific number. Mine. The number.
1 minus 10 to the power of 4.
Right in the chat, in the chat. Thank you. Just give me the power. Give me the power, guys. Give me the power. Balaji, correct. Salih, correct. Tijan, wrong. Muhammad Abu Ghandar. Yes, correct. Give me the power only, guys. Power 1, wrong, Hamad, not power 1. Yeah, Abdullah Malik, Faizan, Diego, Abdullah Barudi are correct all.
Sir, are you chatting to, is the chat to everybody or to host only?
No, to host. Only I see it. Not one. No one see it. Okay. Most of you correct. Actually, it's power minus 4. How? Let's see. Just move, as we said, ya habibi, if you want to convert 0.0001, how to convert it to scientific number? Move the point 1, 2, 3, 4. So it becomes 1 times 10 power, or what, I wrote it like this. 1 10 power minus 4. Got it? So here it's 1 times 10 power minus 4. Okay. Who can give me example on basic? Very quickly in the chat. Expect number, basic number. In the chat. Yusuf, correct. Yusuf Jabari. Tijan, correct. Balaji, correct. Muhammad Bagandar, wrong. Arouk, correct. Faizan, no. Faizan, give me, give me concentration, ya shabab. Don't give me scientific number. Give me number, concentration. Ali Abbas, correct. Farhan, okay. Saleh, okay. Hamad, no. Ali Dakhil, sorry, Mr. Ali Dakhil, charge ya habibi your laptop. No problem.
Hello?
Ali, yes habibi, huh?
Mister, my phone battery died, so I want to bring the charger.
Yeah, bring it quickly. Go, bring it quick.
No, I brought it.
Oh,
That's why I disconnected.
Yeah, no problem. Okay. Vincent, okay. Mustafa. Okay, guys. The idea that if you are talking about basic solution, so the concentration of H plus in this solution must be less than in water. So if in water, we said scientists found in water concentration, this number between the brackets here, okay? So honestly, you can expect what is the basic concentration, H plus concentration. It's supposed to be greater, more than 6 zeros, like this number. If you convert this to scientific number, so move the comma 1, 2, 3, 4, 5, 6, 7, 8, 9 steps. So 1 times minus power 9. Minus 9, sorry. Okay? Okay. So you can use these numbers to describe the concentration of H plus. You can use this scientific number to describe the concentration of H plus. But we have a third way to convert the scientific number for more easier number. It's called the pH number. What is the pH number? pH number, we take, I don't know if you took this in math or not, you have to take the minus log, oh, sorry. You have to take the minus log the concentration of H plus. I don't know, Muhammad Bagandar, did you take this in math, logarithm, or not yet?
Yes,
No,
I
not
think so
yet.
you did.
You did?
No, we didn't.
No, we didn't. No, we didn't.
In grade 8, grade 7?
No,
No.
mister. No,
No.
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 Okay.
Something we never studied.
Okay, until you take it in math, you would find that
Probably next year.
if you take this class in math, you will find that if you take the minus logarithm of 1 times 10 power minus 4, it's 4. Easy, it's easy. Here we are using a small part. If you convert this number, if you take the minus logarithm for this number, you would find it 7. And if you take the logarithm minus logarithm of this number, you would find it 9. Easy, right? So the neutral, the neutral number is 7. If the number raising up, you don't understand because you didn't take this in math. The minus logarithm of the concentration of H plus is called pH. Okay? The pH is converting the scientific number into number. just the power, take it and put it as number. Okay?
Yeah, change the sign and put it as a normal.
Yeah, of course. So here, 4, 7, 9. So whenever, if I tell you pH of this solution equal 9, it means this solution is basic. And this solution in the same moment has 1 times 10 minus 9 concentration of H+. Got it? You got the point?
Okay.
This is the
Okay.
meaning of pH number 9.
Yes.
Okay.
Okay.
Actually, the scale, the greatest.
Yes, I got it. Alhamdulillah.
Yeah, alhamdulillah. The greatest basic, the pH number is, the greatest one, the pH number is 14. Okay? And the lowest one, lowest acidic, the pH
5.
number is 1. Okay? And the neutral, as we said, is 7. Is it clear? Is it clear? Now, in language of pH, we can make the scale in a new style. We can say it's from 1, here 7, here 14. Number 14, from 7 to 14, it's basic. From 7 toward 1, this is acidic. And here, neutral in the middle. Can you tell me which one has more acidic solution? Number 5 or number 3? Write in chat quickly, quickly. Huh? In chat. 3 or 5? Which one more acidic? Which one more acidic? Which one more acidic? Okay, okay.
Mister, what was the question?
The question, if I give you 2 solutions, one has
Mister,
pH.
what was the question?
I'm saying the question. If I give you 2 solutions, one has pH 3, one has pH 5. Which solution is more acidic? pH 5 or pH 3? Yeah, actually most of you correct. Yes. As you move toward number 1, it's more acidic, like this, more acidic, as well less basic, right? And if you move toward 14, more basic. Yeah. If you want to understand why, back to the numbers here. If you move toward the smaller concentration of H+, more basic. If you want to go to the greater concentration of H+, it's acidic. And we converted these concentration numbers into pH number. So as you move from 7 toward 1, it's more acidic. From 7 to toward 14, it's basic. That's it for the pH. I want to move to on the slides quickly. Here, and very quickly, we explain this. The pH scale, this is called, I just draw pH scale. I will give you a chance to ask, guys, but let me finish this. The pH scale indicate the concentration of H+ ions in solution. The pH scale ranges from 0, not 1. I said 1 or 0? What I said there? What I said there, guys?
You said 1.
Oh,
1. You said 1. You said 1.
sorry, 0.
So 0?
Yeah, I mean
Okay.
0. I mean 0. Sorry.
So 0 is the most acidic.
The most, yeah. Do you know what 0 means? That all of it H+, no OH minus. You see, it's very strong acid. But yani,
Can there be -1?
yeah. By the way, I just want to tell you that in your stomach, for example, do you know how much the pH in your stomach?
4.
pH.
No.
3 or
No.
2. 4.
2.
Zero?
2.
Okay,
It's
three.
very, very strong acid. Do you know what do you mean by 2 or 1? If you say this solution has pH 2 or 1, means that if you put it in your skin, it's burn it. Okay? It dissolves your skin. Look how much is very strong acid.
Mister, 7.4.
But
Body.
no, wrong to say this. it's different from place to place in your body. If you are talking about the stomach, inside the stomach, because the stomach produces HCl. So this HCl makes the acidity of, or make the pH of the stomach 2. Okay? Yeah. And when we study the digestive system, you will find that this is important for your body. Number one, it kills the microbes, the germs, which enter with food. When you eat food, many germs enter with the food. The stomach acid kill these microbes. Okay? So it's for protection from microbes. Number two, this HCl provide a good condition for the enzymes which digest the protein in the stomach.
Pepsinogen.
Pepsin, yes, excellent, pepsin. So the enzymes which digest the protein in the stomach is active because of this pH 2. Okay, so
Haragan.
yeah, well, welcome. Wait, just give me a second. Yeah, Muhammad Abu Andar. Another place in the body, like in the blood. In the blood, the pH is 7.4, and this give a good conditions for the enzymes to work there. In your small intestine, for example, the pH is 8. Okay, it's acidic or basic. So when we say 8,
Basic.
basic. But, yeah, but we called it weak basic, or okay, weak basic.
Alkaline.
Or alkaline. Yeah, right. Okay, guys, excellent. So the pH scale. This is the pH scale. You can see it like this. It's numbered from 0 to 14. And here we have some examples. The pure water, not this water which you drink. This water which you drink could be as weak, acidic, or basic. Not pure, not neutral, because it's not pure. It has some dissolved minerals and other stuff. The pure water, if you remember in the lab, do you remember in the lab when we used a water, we made it in the machine? You remember?
No.
In the water bottle, this
Yes,
water
the
called,
Benthic.
yeah,
The
this called,
Benthic.
excellent. This was called distilled water, right? This water is a pure, only H2O, nothing there else. Okay. What I want to say, example, human blood, 7.4, as we said. Human stomach, if you want to add it, it's here. Human stomach. Okay. Seawater, alkaline, you see, or basic. Time. Yeah, here, look, examples, another examples, tomato juice, lemon juice, stomach juice. And last year, and next time, inshallah, we will make a virtual lab about acidic and basic and see how it's.
Mister, you said that the stomach acids produce 2 pH, but over here it's saying 1.5.
pH 2. It's a range,
Mister.
yeah, Diego. It's range between 1 and 2. It's range. It's not specific to 1.5.
Mr. Basim,
Yes.
are the stomach acids what help the metabolism to increase?
Metabolic acid? What
No, are the stomach acids
Yes.
or the acid or
Yeah.
the high acidic in the
Yeah.
stomach what helps the metabolism? The metabolism?
No, it just activate the enzymes, so that increase the digestion of the food, not more. Okay. Here are other examples of pH more than 7.
Mister,
Yes.
I was trying to open the PPT today, not yesterday. I did not open the PPT. Like when I used to do the continue thing, and it wouldn't open at all.
It's not open. Not open. Not open the file.
No.
Okay.
No, any of the PowerPoints.
Does not open.
Yeah, it's not
No.
Okay. I will send it to you again. Don't worry. I will send it to you again, inshallah.
Okay.
Okay. And that's it, guys. And everything here explained. We just, yeah. The last thing, and we finish, something called buffer. What is the buffer? Buffer is a chemical that maintain the pH. What do you mean by maintain the pH? Here in your body, we just said that the stomach pH must be between 1 to 2, not greater يعني, not more than 2. Okay, so how the stomach maintain the pH while we already eat food? Sometimes this food has OH minus, which decrease the H pluses in the stomach, so decrease the acidity, right? So here the buffer work. What is the buffer? And even in the blood, how the blood keep the pH 7.5? What if you drink lemon? What if you drink lemon juice? And this lemon juice, you know, absorbed by the blood. Does this affect on the acidity of blood? We say no. How come? The mister just said that if you add for anything has H plus to the solution, so that will increase the H pluses in the solution, so it becomes more acidic. That's what we learned, right? So if you drink lemon juice, which has more H pluses, so that would increase the concentration of H plus in the blood, so makes the blood more acidic. This is the logic. But in reality, the pH of blood does not change. Oh, how come? Actually, there's a something in the blood. There's a something in the blood. It's
Buffer.
work. Yeah, it's called buffer. It's a chemical. This buffer, this compound, its function that decrease the H plus if it's increase. Means that the H pluses which come from the lemon juice, for example, will be removed by the buffer. Okay, what if you drink something, decrease the H plus in the blood? So the buffer will add H plus to the blood. So it's like balancing. You got the point? Balancing the H plus in the blood. Let's see in very quickly way, because the time is going to finish. Here, this is blood vessel, H plus, H plus, H plus, H plus, for example. Now, if you drink something, increase the H plus. Okay, increase the H plus in the blood. Let's change the color. So that would increase the H pluses in the blood, so the pH will be more acidic, right? And we don't want this to happen. How? The buffer will take these two H pluses after they enter the blood. Let's see what is this. Actually, this is the HCO3 minus. This buffer can react with H plus to form H2CO3. That's it. Okay, again, HCO3 minus, this compound able to react with hydrogen ion to form H2CO3. You got the point? And it can do the opposite. This H2CO3 can break down into H plus and HCO3 minus. So how to maintain the concentration of H plus? If the food, for example, increase the H plus in the blood, which reaction will happen, from left to right or from right to left?
Left to right.
Left to right. That this HCO3 minus will cancel some H pluses to keep the concentration same, not changed. You got the point? Okay.
Yes, Mr.
Okay, what
Basically
if,
lower it.
yes, what if, another example, what if the H plus in the blood, we just said maintain to 7.4. What if you eat food, produce OH minus, so that decrease the H pluses as we explained before, to make it more basic, right? But we don't want this to happen. How?
Yeah.
The H2CO3, the H2CO3, sorry, not minus, splits into H plus and HCO3 minus. Why? To give this H plus to the blood, to replace the lost H pluses here. Got it? So by this way, the blood kept his pH same, 7.4. Or in different words, kept the H plus concentration same. Easy game, right? Right?
Yes.
Can you repeat again? So can you repeat again?
Okay, I will repeat it, but that's it. I will repeat after a while. That's it about the pH, guys. The buffer maintained the pH by this way. And inshallah, we will have a lab, virtual lab, to explain how this buffer work, okay? Within few seconds, we want to answer on the chat, please. This quiz, yalla, quickly, huh? Yalla, quickly, on the chat, give me the answer for the first one. In the chat. A molecule in which the charge are unevenly distributed is a call of? Answer is, oh, all of you correct. Excellent. A. Okay, question number 2. Please write the number of questions beside your answer. Question number 2. A dissolved substance is called a dissolved substance. Wrong. Who said B? A dissolved substance is called a dissolved solid. Solid is the dissolved things. The substance which dissolve in the water or in the solvent is called solid. Okay, number 3, question number 3. Yalla, question number 3. A compound that produces a hydroxide ions in solution is called a hydroxide ions. You know what hydroxide ions? OH minus.
Hydrogen ions.
OH minus. No, not hydrogen ions. Hydroxide ions, OH minus. So any compound increase the OH minus to the solution. This is called, what we call this compound? Answer is base. Guys, the compound which the people who said buffer, it's wrong. Buffer does not only increase OH minus. Buffer increase buffer does not increase OH minus. Buffer increase or decrease H plus only. Okay, you got the point. In this example, in the blood buffer, for example. But here I just choose, or the question asks for compound only increase OH minus or produce OH minus. So it's a base. Yalla, number 4, quickly, huh? Question number 4. Hydrogen bonds between water molecules results from? Why there are hydrogen bonds between the water molecules? It's easy. Okay, I see different answers. Why? The hydrogen bonds between water molecules results from water molecules attract to each other. Why? The H2O attracted to the H2O. H2O, sorry. Balaji correct. Ali Rusan correct. Salih correct. And the others, I think no. The answer is C, uneven. We said water molecules polar, carry charge due to uneven electron distribution in each water molecule. So they are charged. That's why the molecules are attracted to each other. The next one and the last. On pH scale, a value of 2 means that the solution has, yalla, quickly. Oh, different answers I see. C, 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 D, D, C, D, C, C. What is this? No. Look. C. People who said C, you are correct. A higher concentration of H plus than pure water. This is a value 2. BH2 means more acidic, more H pluses, more H plus concentration. Thank you for that. Sorry for because we took some of your break time, part of your break time. And okay, guys, you're gonna find a worksheet on Classera. You have to do it and submit to me. Thank you very much. You can leave now. If you have any question, stay.
Bye, Mister.
Mister, when do you
Bye,
submit it?
mister.
When will the worksheet be
Bye,
uploaded?
bye, bye.
Bye.
When
Mister,
will the
when do you
worksheet be
submit the
uploaded?
worksheet?
I'll be making ads for restaurants in your class.
Yalla, if you want, anyone want a question?
Mr.
Sir,
Bassel?
when will
Who
the
wants,
worksheet
who
be
wants
uploaded?
question? Who want to ask? Wait,
Yes.
just a second. If you don't want to ask, you can leave, guys. If you have another class or something. Yes, yeah, Ali Rosan. Yes, speak. Huh?
The worksheet, when do you submit it?
The worksheet on Classera.
No, when?
When?
Due
Tomorrow.
today?
Due time.
Okay.
After I submit it by 1 day. So
Okay.
tomorrow.
Okay.
Tomorrow, because today, inshallah,
Bye,
we'll
mister.
submit it. Bye bye.
Salam, Mister.
مع السلامة. Any question
Mr.
else?
Bassel.
Habibi.
I have a question.
Yes.
So can you explain the buffer thing again? The buffer molecule.
Yeah,
What
buffer
is
was
it?
confusing.
What is it? What is
Buffer.
it?
The buffer.
Buffer. Okay.
The
Yeah.
example, yeah, the example you gave.
Okay. The buffer I gave you example about, we just said the buffer, we just said the buffer that a compound maintain the pH. How maintain the pH? By keeping, keep the concentration of H plus in a range. Means that do not decrease or increase the pH. Do not allow for increase or decrease in the H plus concentration in a solution.
Why?
Why? Because, for example, in the blood, or we just said that there are enzymes, proteins work in the blood under this pH 7.7.4. If the pH changed, that's affect on the work of enzymes. And inshallah, when we study the enzymes structure, you will find that change the pH damage the enzymes. You understand? So that's why the pH in the blood or the pH in the small intestine, the pH in the stomach, supposed to be within its range, does not change. Like in small intestine, the pH is 8, do not change. It's supposed to stay in range, do not change out of 8. You got the point? The pH of blood 7.4 supposed to be 7.4, not to change, not allowed to change. Why? We just said any change in the pH that damage, or in biology we call it denature the enzymes, damage the enzymes. Okay, you got this point now? So that's why the in the body we need something to maintain the pH, to not change. But the problem that we eat some things change the pH, or we drink drinks change the pH. So how to maintain the pH? This is the work of buffer. The buffer do like this. Look how buffers do. Now we just said that you have a specific, for example, in small intestine, let's say, in small intestine, the pH supposed to be 8. So there's a specific concentration of pH. Do you remember? 110 minus 8. This is the concentration of H plus in the small intestine, or pH 8, you can say. Now, if you drink lemon juice, lemon juice is acidic solution. Means that it has much of H pluses. We just say it, it's strong acid, has much of concentration of H plus. So that will increase the H plus in the small intestine. Increase the H plus in the small intestine will change the pH. And we don't want this to happen. What to do? Not to drink lemon juice. This is not a good solution. We need another solution. We need another idea. It's the buffer.
The buffer will maintain the H pluses.
Yeah, buffer.
And it'll keep it in range.
Buffer watching or observing the small intestine solutions. if it finds that the H plus is increased due to drinking lemon juice, orange juice, or whatever, so directly what the buffer does, catch these H pluses, catch it and take it to remove its effect. We know that
What
increase.
What does
with
it do
it?
with it?
I will
What does
tell
it
you.
do with it? Yeah.
I will tell you. The H plus, if it's increased, the effect, change the pH. So remove the H plus from the solution in the small intestine, remove its effect. You agree with me? Agree?
So basically,
Yeah,
it
yeah, yeah.
balances
Yeah.
like the balancing equations. So basically,
Second, give me
balances the
second, give me
molecule.
second, give me second. Stay with me step by step because of the time, 3 minutes left. Now, we said that if you remove the H pluses, the X's H pluses, so the pH will not change. You agree with me? You agree?
Yeah, yeah.
Okay.
Yes.
Okay. So how the buffer remove the H plus? Easy. The buffer is HCO3 minus. This is the buffer. Is a compound. This buffer, HCO3 minus, able to react with H plus and becomes H2CO3. So no H plus anymore. Got it? So many of HCO3 minus there found there, they're working on binding with H plus. Okay, if there are X's H plus. But what if the opposite? What if
Mister, I have a question. Like
wait,
what?
give me wait because we have less than 2 minutes. Wait. What if the H pluses? Now we just said the pH is 8 due to a concentration of H plus or hydrogen ions in the small intestine. What if you eat something produce OH negative? So that makes it more basic. We just answered a question in the quiz. More OH minus that decrease the acidity. So change the pH. And we don't want this to happen. But before, how the OH minus change the pH? The OH minus react with the H pluses as we explained in the beginning of the class and decrease it. So the H pluses will be decreased due to the OH minus which enter by food, for example. So how the buffer does to return back the H plus concentration to the pH 8, the buffer now works like this. H2CO3 splits into H plus and HCO3 minus. And this H plus added to the solution in the small intestine. So the red H pluses replaced the cancelled H pluses by the OH minus. Is it easy?
Sebastian, I have a question.
Quickly, because we have less than a minute. Quickly, huh?
Yes. You
Yes.
said this reacts with this, right?
Yes. Yes.
Yeah. Why does this not react with this? Why not react with this? Why does it react with this?
It's same. It's same. And this I gave it two different black and blue just to make it easier for you. No matter.
No. I mean, no, I understand. But then
I want the concentration same, whatever this or that. No matter. The matter is keeping the concentration of H plus same. The X is H plus we don't need it. The buffer, remove it.
how does it know if it's X?